We each have a chronological age, which is simply the time that has passed since birth, but our brains may have a biological age of their own. It's now possible for scientists to estimate this brain age from medical scans, and this metric can provide significant insight into an individual's health. In this explainer, we will examine a large-scale study that investigates the genetic basis for why some brains appear to age more rapidly than others.
To understand this research, we'll first define what biological brain age is. Next, we will cover the genetic associations uncovered by the study. From there, we'll explore how brain age is connected to overall health. Then, investigate which factors might causally contribute to accelerated brain aging. And finally, we'll consider the implications of these findings.
So, let's begin with the primary measurement used in this field. The biological age of the brain is assessed using a concept known as the brain age gap.
The key metric is the brain age gap, often abbreviated as BAG. This is calculated by first training a machine learning model on thousands of brain MRI scans to predict a person's age based on brain structure. Then, a new individual scan is analyzed. If the model predicts an age of 45, but the person's chronological age is 40, they have a positive brain age gap of 5 years.
This gap is clinically relevant. When a person's brain appears older than their chronological age, a positive brain age gap, it's associated with a range of negative health outcomes. These include reduced grip strength, elevated blood pressure, poorer cognitive performance, depression, and an increased risk for neurodegenerative disorders such as Alzheimer's disease. The association between an older-appearing brain and adverse health raises a fundamental question: what determines this gap? Is it primarily lifestyle, environmental exposures, or genetics? Previous research has suggested a substantial genetic component, which is the specific focus of this investigation. To identify these genetic links, the researchers performed a large genome-wide association study, or GWAS. This method involves scanning the entire human genome to find small variations that are associated with a particular trait, in this case, brain aging. This study represents the largest such effort on this topic to date. To achieve sufficient statistical power, data from multiple cohorts were combined, resulting in a total sample size of over 56,000 individuals. A sample of this magnitude allows for the detection of subtle genetic signals that might be missed in smaller studies. The analysis was productive. It identified 59 distinct locations in the human genome, referred to as loci, that are significantly associated with the brain age gap. These can be considered 59 genetic regions of interest for brain aging. What is particularly noteworthy is that a majority of these findings are novel. Of the 59 genetic loci, 39 had not been previously linked to brain aging. This represents a considerable advance in mapping the genetic architecture of this trait, enabled by the study's scale. This raises the question of how much of brain age is determined by genetics. The study estimated its heritability. As the chart illustrates, common genetic variants are estimated to account for approximately 23 to 29% of the variance in the brain age gap. This indicates that the remaining proportion is attributable to other factors, such as environment, lifestyle, or less common genetic variants. Looking more closely, the researchers prioritized specific genes that likely underlie these associations. Some were well established in neuroscience, such as MAPT and APOE, both of which have strong links to Alzheimer's disease. They also identified other candidates, like the DPF3 gene, which is involved in the generation of new neurons, and TNIK, which is connected to general aging pathways. The single most significant genetic signal was detected on chromosome 17, in a region that contains the MAPT gene. This is a powerful finding as it reinforces the hypothesis that the processes of typical brain aging and the pathology of diseases like Alzheimer's may share common biological mechanisms.
We've established that genes influence brain age. The next step was to determine if these same genes affect other aspects of health. To do this, the researchers compared the genetic profile for brain age with the genetic profiles of more than 1,000 other traits, including diseases and socioeconomic factors.
The results showed clear patterns of genetic overlap. The genetic variants predisposing an individual to an older-appearing brain were also associated with higher blood pressure, diabetes, and depression. Conversely, the genetic variants for a younger-appearing brain showed overlap with factors like higher educational attainment, higher income, and longer parental lifespan.
The key insight here is that brain aging does not appear to be a genetically isolated process. The same genetic architecture that influences the rate of brain aging also seems to be intertwined with broader aspects of an individual's health, affecting cardiovascular, cognitive, and mental health, and even socioeconomic outcomes.
It is important to remember that correlation does not imply causation. Shared genetic influences do not prove that one trait causes the other. To test for causality, for example, to see if high blood pressure actually causes accelerated brain aging, the researchers employed a specific statistical method.
This method is called Mendelian randomization. It functions as a form of natural experiment. Because genetic variants are randomly assigned at conception, we can use the genes associated with a risk factor, like high blood pressure, as an instrument to study the effect of that risk factor itself. This approach helps to minimize confounding from lifestyle factors that often complicate observational studies.
Let's use blood pressure as an example of how this works. First, the analysis identifies individuals with a genetic predisposition to higher blood pressure. Second, it tests whether those same individuals also tend to have a larger brain age gap. The analysis found that they do. This provides evidence that higher blood pressure has a causal effect on brain aging, rather than being merely correlated with it. Using this approach, the study provided evidence that a few key health factors are not just correlated with, but appear to be causal risk factors for accelerated brain aging. Specifically, both higher systolic and diastolic blood pressure, as well as type 2 diabetes, were implicated. This is a significant finding because these are modifiable risk factors.
So, what are the broader implications of this research for our understanding of brain health? Let's consider what these findings might mean for future work in this area.
One final question relates to the genetic complexity of brain aging, a concept known as polygenicity. The researchers estimated that approximately 11,000 common genetic variants likely influence this trait. As you can see from the chart, this level of complexity is comparable to a physical trait like height and lower than that of a behavioral trait like neuroticism.
This moderate level of polygenicity is promising for the field. It suggests that identifying a substantial portion of the remaining genetic variants that contribute to brain aging is a feasible goal for future, even larger studies.
To conclude, as the study's authors state, their work helps to refine our understanding of the genetic architecture of the brain age gap. By identifying new genes and pathways, this research provides a foundation for subsequent work on the biological mechanisms of brain aging and the identification of potential therapeutic targets. So, this study provides a more detailed genetic map for brain aging and confirms that manageable conditions like high blood pressure and diabetes can causally accelerate it. This leaves us with an important forward-looking question. Can this improved genetic understanding be leveraged to develop targeted interventions that effectively protect and preserve neuronal health over the lifespan?